morning, Mr. Chang and fellow classmates. We are Group 4, Science Works 1, 2, 3, and today we're presenting to you our science experiment. I'm Ying, the group leader, and these are my fellow group mates, Rui-En and Jermaine. Our topic for our science project is biology, and particularly studying about the effects of the exposure of UV light on bacterial growth. Our aim of the experiment is to find out if the longer amount of time exposed to UV light will result in decreased growth of bacteria. We knew through prior knowledge and some research that bacterial growth is affected by factors, namely temperature, humidity, light, and light. Our main focus of today, however, will be on the factor of light, because UV light is one of the components of sunlight. It is also known that UV light prevents the growth of bacteria from mutating the cell. Thus, based on this knowledge, we hypothesized that the algae plate that is exposed to the longest duration of UV light, which is 9 minutes, we have the least amount of bacterial colonies, followed by 6, 3, and 0 minutes. We will then conduct an experiment to prove our statements validity. Now I'll hand the time over to Jermaine, who will be explaining the procedures of the experiment.
Hi, I'm Jermaine, and today I'll be talking on the procedures done to carry out the experiment. Firstly, the apparatus and the materials needed are 12 LB agar plates, a hood, 10 ml E. coli 1011 CFU per ml, and incubator, stopwatch. You would see some of the apparatus used in the pictures in the videos provided later. We also need parafilm for ensuring the samples are tightly sealed, PS membrane used for filtration, a permanent marker, gloves, masking tape, 70% ethanol solution for disinfecting the area, and a long sleeve shirt. These are the procedures to expose the LB agar dishes under ultraviolet light. Firstly, put on the gloves and long sleeve shirt or jackets. This will prevent contamination of the samples by bacterial dead skin cells from our exposed skin. This precaution also prevents us from being exposed to some of the ultraviolet radiation, as excessive amount of radiation can be harmful to human skin. Secondly, turn on the hood and switch on the light and the blower built inside the hood. This is the first step of the sterilization process of the hood to prevent any bacterial contamination by blowing up any floating microscopic organisms. After 5 minutes, spray 70% ethanol solution on the surface of the hood and wipe it with a clean cloth. Ethanol solution helps to disinfect the tabletop, which we'll be doing in our experimental setup. In order to ensure that the environment is completely sterilized of microbes, close the hood and turn on the UV light for 10 minutes for the final stage of sterilization. Now we can finally begin our experiment in a conducive environment. Using a stopwatch, put in 3 LB agar dishes for each time and remove after 9 minutes, 6 minutes, and 3 minutes respectively. Package all the petri dishes using parafilm to prevent the dishes from opening up accidentally and being exposed to the air. Incubate the bacteria overnight in an incubator. The optimum temperature for E. coli is 37 degrees Celsius. E. coli bacteria is used on our experiment as it's a relatively simple model to conduct our experiments and develop our results on. Now I pass the time over to Rui-En, who will continue explaining the experimental procedures. Rui-En, please.
Thank you, Jermaine. Moving on, the procedures to count the colonies of E. coli growing on the LB agar after incubation. As it is impossible to individually count the E. coli colonies growing on the petri dish, we decided to do an estimation. We took an area and estimated the number of E. coli colonies growing on it. We then multiplied it by 4, as the area we had taken was 1/4 of the petri dish. Then we entered the results into our table, including the control, which were the dishes exposed to 0 minutes of UV radiation. We also calculated the standard deviation, which would be shown later. Now, we will show you a video of the apparatus required. These are some of how the apparatus look like. These are the latex gloves that we used in order to protect our hands, and the long sleeve jacket that we wore. This is a 70% ethanol solution that we use to disinfect the area, and the hood and the incubator. These are the petri dishes. This is the PAS membrane filter that we use to filter any unwanted contaminants from our E. coli solution. This is the UV hood which we expose the dishes. This is the parafilm that we use to package the things. And here's another video on some pictures during our procedure. This video also includes the results of our experiment. This is the streaking of bacteria where we use a micropipette to transfer the solution from a container into the petri dishes before exposing them into the UV light. This was done inside the UV hood. We had already sterilized the UV hood with the 70% ethanol solution by wiping it on all surfaces. And this shows the packaging of the parafilm of the Petri dishes in order to ensure that the samples do not accidentally open up. This is a UV hood and its model and information. These are our results after incubation. As expected, the Petri dishes that were exposed to 0 minutes of UV light had the most bacteria growing on them, and the Petri dishes exposed to 9 minutes of UV exposure light had the least bacterial colonies growing on them. Now, Yingjing will take over to elaborate on the results of the experiment.
The results are that the algae agar plate exposed to the shortest amount of UV light, which is 0 minutes, had the most bacterial colonies growing on them. These samples serve as our control setups. The results show that the natural growth rate of E. coli without being exposed to UV light. These are some pictures of our algae agar plates. These plates are the ones that are exposed to 9 minutes of UV light. You can see that it's very little bacterial colonies growing on it, and it can be considered negligible. While these are the algae agar plates that are exposed to 6 minutes of UV lights, there is a moderate amount of bacterial colonies growing on them. Hence, we can conclude that the duration of UV light exposure does affect the growth rate of the bacteria from our data obtained. Now, we will continue with the experiments and results.
Thank you, Yingjing. These are the end results to our experiment. You can see that the algae agar plates with 0 minutes of exposure of UV light had a mean average bacterial colonies of 130, having an STD of 62.4, while the one with 3 minutes had an average of 31.3 bacterial colonies going on it, with an STD of 1.53 followed by 9 minutes, with an average of 0 bacterial colonies and an STD of 0. This slide shows our results compiled into a line graph. The x-axis is the duration of exposure the x-axis is the duration of exposure to algae agar in terms of minutes, and the y-axis is the mean number of bacterial colonies going on the algae agar plates. It is shown here that there is a relatively steady and gradually decreasing number of bacterial colonies as the duration of UV light exposure increases. The trend between the number of bacterial colonies found growing on the algae agar plates and the UV light and the duration of UV light exposure can be thus concluded as such. Next, we will be moving on to the discussion sector of our presentation. To combat the problem of the cyst falling out of our gloves, we can improvise by fastening a rubber band over the gloves. This would lower the chance of the gloves of the end of the sleeve from coming out. The experiment could also have been repeated with small time intervals to more closely inspect the steady change of the bacterial growth. It would also enable us to more closely inspect the steady change of the bacterial growth. This will also help us to ensure that there are no discrepancies in the relationship observed with our gathered data. We could also reschedule our experiment for another day to completely redo the entire procedure so as to prevent human error and ensure that our results were accurate and reliable. We could have also handled the apparatus and materials with more diligence and care so that the algae agar would not be accidentally contaminated, rendering our whole experiment useless. We are aware that whenever preparing cultures for the samples to grow, sterility is of utmost priority. Moreover, additional secondary research could also be conducted to fill us up on prior knowledge so as to better conduct the experiment and know what to expect. The knowledge could also aid us in battling certain loopholes in our procedure and further reduce the error we could make. We could also try to look for online reports that are more recent To ensure the information is reliable, we could set a specific time frame, which, for example, could be like the report has to be last updated not more than a year ago. Lastly, gathering more information from our teachers and mentors could also be a massive help to the experiment. Next, Jermaine will now take over and conclude this presentation.
Okay, so our conclusion is that our hypothesis, which is that a longer exposure of bacteria to UV light will result in a decreased bacterial growth, which is shown to be correct. Our result tends to agree with our literature review, being that UV light is able to mutate the cell and disable it from reproduction. This proves that UV light does, in fact, help to control the growth rate of bacteria. These are the references and credits that has helped us greatly in our research for the topic for this science project. For our acknowledgement, we would like to thank Mr. David Chung for his constant guidance and Tae Rong Eun for giving us his valued feedback on this science report. We have come to the